Okay, so that brings us to the dendrites, the other region of the neurons. And so what do the dendrites do? So if the axons transmit signals, then it follows, or it's convenient anyway, that the dendrites receive signals. And they do this not through the use of action potentials, but instead through graded potentials. So we've talked about graded potentials already. They are basically what is not an action potential. Anything that's not an action potential we'll call a graded potential. And so they're not all or none. They can be a variety of magnitudes, different membrane potentials. And we'll see how that is necessary given the functions of the dendrites. Okay, so they are going to transduce stimulus via electrical signals, and they're finally going to decide whether or not to generate an action potential. It's really the junction between what we call the dendrites and the axon where this decision is made, is sort of the intermediate zone between them. And because of that zone, this decision about whether or not an action potential is generated, because of that, we have the opportunity for a single cell to make decisions. That is the stimulus that I'm collecting, is this information that I'm bringing together into the cell via all these branches in the dendrites, is that enough to transmit a signal along my axon? And so we have the opportunity for integration of signals in this cell. So those are the topics that we're going to talk about. We're going to pick up where we left off at the chemical synapse. And we're in this space between the presynaptic and postsynaptic cells. It's called the synaptic cleft. We're zoomed in up top there. We talked just a moment ago about that axon, and now we're talking about this finger-like part, the dendrite on the postsynaptic cell. And we use the example of acetylcholine, so we'll stick with that. It's a neurotransmitter that's released by the presynaptic cell. And when that presynaptic cell is activated by an action potential, we have a release of lots of acetylcholine via lots of vesicles that bind to the plasma membrane of that cell. And so we have all that acetylcholine released, free to diffuse down its chemical concentration gradient. And on the postsynaptic side, if that cell is receptive to that neurotransmitter, then it will bind to ligand-gated ion channels. Okay, so we talked about voltage-gated channels. I mentioned that there are such things as mechanically gated channels. We'll talk about those later a little bit. This is a ligand-gated channel. So it's going to bind to a chemical. In this case, it's specific to acetylcholine. That causes a gate to open and for ions to move across the membrane. So by binding to that chemical, we have the opportunity to tweak the parameters in the Goldman equation, in particular that permeability in the Goldman equation, so we can now drive the membrane potential towards the equilibrium potential for whatever ion this channel is sensitive to, or I should say permeable to. So in this case, we're going to go with sodium. So it's a ligand-gated sodium channel, and when it opens up, sodium fluxes into the cell, and we have what? A depolarization. Just like we saw for the axons, it's just that in this case, we're not going to create an action potential. We're just going to create a positive change in the membrane, in other words, a depolarization. So we show that in red. Okay, so this would be an example of an excitatory stimulus. Excitatory synapse. So a particular chemical synapse, that is one particular junction between cells, is either going to be excitatory or inhibitory. And so you're either going to have a whole bunch of ligand-gated sodium channels, which would be the case for the excitatory version, or a whole bunch of ligand-gated chloride channels. And we'll get to that in a moment. So we're used to thinking about sodium as doing its excitatory thing, because it's exactly like what function it serves in the axon. If this was instead maybe at a different site on the dendrites of the same cell, we might have a different chemical synapse, and that chemical synapse might have voltage or ligand-gated chloride channels on the postsynaptic cell, on the dendrites there, so that when they bind a neurotransmitter, they allow chloride across the membrane. And that, it turns out, so chloride is moving in the same direction as sodium. It's more concentrated outside the cell, but it has the opposite valence as sodium. And so as a consequence, it has an equilibrium potential that's not positive like sodium, but it's actually negative. And we see that equilibrium potential right here, this value here. So we use the Nernst equation. If we know the concentration difference, we can arrive at this number, pretty close to the equilibrium potential for potassium. So chloride, despite the fact that chloride is negative, potassium is positive, they both have a similar equilibrium potential. And their concentrations are similar as well. So the bottom line is, if your chemical synapse has ligand-gated chloride channels, that's going to be an inhibitory synapse because it's going to tend to take the membrane potential and move it from -70 towards this more negative number, which means that it's going to hyperpolarize the membrane. It's going to make it more negative and therefore less likely to be activated by an excitatory signal. All right, so we call this an inhibitory synapse. It's a hyperpolarization that's created by ligand-gated channels that increase chloride permeability. Not chlorine, some people say chloride. Okay, does that make sense? This is the first appearance that we've seen, aside from the Goldman equation, the first time that chloride actually matters, and we see that it matters in the context of providing an inhibitory synapse. Question?
Actually, a comment. So all we need to worry about is, so Na plus depolarization, hyperpolarization PL-, basically, via ligand-gated channel, right?
Well, I wouldn't say that's all you need to know, but you need to know that. Yeah. Okay. In a different chemical synapse, we might have what are called G-protein coupled receptors. And the G-protein coupled receptors are a whole large class of transmembrane proteins that serve a variety of functions. So we're talking in intentionally vague terms because there's really a whole universe of really interesting cellular physiology. But for our purposes, it's relevant because they provide the opportunity for long-term changes to the postsynaptic cell. This is all happening at the dendrites. So if they bind to a ligand that excites this G-protein coupled receptor, then they may cause a variety of intracellular actions called second messenger pathways, which may influence the state of that membrane. So, for example, and they can do things like initiate transcription, they can activate enzymes inside of the cell, they can cause a variety of intracellular changes that are then reflected in the membrane potential. So if a second messenger pathway, for example, influences the open or closed state of a channel, then they can influence the membrane potential. So we'll say that in general, these second messenger systems are pathways that transmit information within the cell. And in the case of neurons, they can increase or decrease the open state of ion channels. They can also change the presence of leak channels. And so potassium leak channels, for example, they can allow for those leak channels can be sequestered in the interior of the cell in vesicles. And second messenger pathways can result in those channels binding to the cell membrane and therefore effectively making the membrane more leaky to potassium, increasing its permeability. So, so what this introduces is the possibility for long-term changes to the membrane potential. So if it's a long-term depolarization, that means that the cell is going to be primed for an excitatory stimulus. It doesn't require much of a stimulus from a presynaptic cell to get that cell to create its own action potential. Whereas if it tends to be hyperpolarized, it's going to require much more of a stimulus to activate that cell. Okay, so it's sort of like modulating the response to other stimuli by these long-term changes. And they can have a variety of other downstream effects. Okay, so again, second messengers is a little fuzzy, but I just want to introduce the notion that you can have these more complex long-term changes to the membrane potential. So that brings us to a clicker question, which is about these second messenger pathways. What would happen to the membrane potential if a second messenger reduced the number of potassium leak channels on the membrane? Would it be nothing? Would it would B, hyperpolarize the membrane, or C, it would depolarize the membrane? Okay, so take a minute to think about that. Okay, give me your final answer on this one. Final answer. Okay. So we're changing, so leak channels function to enhance permeability, right? So they're gonna, in this case, enhance the permeability for potassium. So what that means is that they are going to cause the membrane potential to move towards the equilibrium potential for potassium. That's what they do. So here we have a scenario where we're going to reduce the number of leak channels and therefore reduce the permeability of the membrane to potassium. That means that at rest, we started off at a point where we were close to that equilibrium potential. We're going to reduce the permeability, so we're going to make potassium less important in its influence on the membrane potential. So it's not going to be nothing, and in fact, it's going to tend to depolarize because potassium, again, keeps it hyperpolarized. If we reduce the influence of potassium, then we're going to move away from that. All right, so much like when we talked about how it was critical for action potentials that the membrane reset quickly so they can receive another signal. The same is true for a chemical synapse. If we're receiving some excitatory or inhibitory signal from a presynaptic cell, in order to be receptive to a series of signals, a train of action potentials, we need to clear out the neurotransmitter very rapidly. So this is done a few different ways. First of all, this is just a space, an interstitial space between cells, and so the neurotransmitter will just sort of diffuse away from the cells and be lost. The second is that there are proteins that transport the neurotransmitter from the cleft and transport it back into the presynaptic neuron. And finally, we have enzymes that break down the neurotransmitter so that they're not activating any of the ligand-gated channels on the postsynaptic side. Okay, so that's just a little story about how the neurotransmitters are moved rapidly so that we can receive another signal on the postsynaptic cell. All right, so more sort of critically, let's talk about how an action potential gets started. Okay, so because this relates to the function of the dendrites and a neuron in general of integrating information and making decisions. So let's consider the case of an excitatory synapse. So we have a presynaptic cell, we have this little cleft, and on the postsynaptic side, we have voltage or ligand-gated sodium channels that allow for the diffusion of sodium into the interior of the cell upon release of that neurotransmitter. Now that, again, is going to create a depolarization. And if that depolarization is enough to get the voltage-gated sodium channels, that is the voltage-gated sodium channels in the axon to reach threshold, then we're going to create an action potential. Okay, so we call this bottleneck region, it's an anatomical term, the axon hillock. But since that's showing us where the axon begins, we know that axons have voltage-gated channels. And so that hillock is known as the spike initiating zone because that's where we have the opportunity to generate spikes, which is another term for an action potential. So the spike initiating zone is really where the decision is made about whether or not an action potential is going to be generated by this cell. Oops, sorry. Okay, so it's at the base of the axon, and it's the site where the action potential begins. All right, so this might seem like, what does it matter if an action potential is generated? Well, through networks of neurons, the way that they make decisions and the way that they store information, transmit information, is about this action, right? Whether or not a signal gets transferred from one cell to the other. So this property of the voltage-gated channels being activated or not activated provides that opportunity. It's like the ones and zeros that computers use for transmitting information and storing information. It's just a binary difference that has, when you look across a more complex network of these cells, it's a critical property that allows for a lot of complexity. Okay, so let's interrogate this a little bit further. So we're going to do a little virtual neurophysiology. We're going to monitor the membrane potential right in the vicinity of that chemical synapse in red. We're going to look downstream the dendrite, and then eventually we'll look beyond the spike initiating zone. We're going to color code that, and for each site, we'll monitor the membrane potential and look as a function of time. Now, initially, this is a chemical synapse, and so you should expect a depolarization. And we see one that looks a lot like an action potential. It's not, but it looks like it. And we know that it's not because when you look a little bit later, you see that magnitude of depolarization is not maintained. It declines. So that means we're dealing with graded potentials, much like on the inside of the myelination. And as you look further on, you keep losing that positive change created by the excitatory synapse. In the event that that depolarization is sufficient to go above threshold, we have a depolarization that's quite rapid. In fact, it's an action potential. So again, the dendrites don't have voltage-gated channels and therefore can't create an action potential, whereas the axon does. Okay. So here we see an example of a single excitatory synapse passing via an action potential neurotransmitter to the postsynaptic cell, causing an influx of sodium, driving a depolarization, and that is enough to get a spike in the axon. So the rule here is that action potentials are created if the membrane potential is above threshold at the axon hillock, exactly what you would expect from what we discussed already about axons. All right, so let's consider the scenario where this excitatory synapse is not so strong. So we get a relatively minor depolarization. Maybe there aren't as many ligand-gated channels on the postsynaptic cell, and we get a small depolarization. And if that's so small that it doesn't reach threshold, then we just lose all that information. That is, this cell does not transmit an action potential. The information is essentially lost. thank you, but that's just not useful. The information isn't transmitted. So not all synapses create an action potential. Okay, so how can we help to ensure that the information is transmitted? What can be done to weigh in a way where we favor the generation of the action potential? Well, one thing you could do is, instead of just sending one action potential and one release of neurotransmitter, what if you sent a train of them, or in the simplest case, just two depolarizations? So two depolarizations, we can see that in the dendrites, we have the opportunity for the membrane to behave a little bit differently from an axon. So for an axon, you're either generating an action potential or you're like recovering from the beginning of the action potential. So you're in the absolute or the relative refractory periods, and it's only when the membrane has reset or is close to it in the relative refractory period that you can then get another action potential. But here we have something that's different, because we can see that there's two excitations here. Again, we're dealing with a excitatory synapse. So on the first release of neurotransmitter, we get this depolarization. On the second one, we get one that's greater. And in between, we actually don't return to the resting membrane potential. So that's kind of different, because we're used to thinking of these membranes as resetting and being ready to accept another signal. Here we see that by sending a pair of action potentials, we're kind of like building a depolarization. And that is possible with graded potentials. There's a release of sodium, and before the membrane can recover from that, there's another release of sodium. And as a consequence, that second action potential has a stronger effect than the first one does. So the history of these action potentials and the timing between them matters to what membrane you can generate on the dendrite, or what depolarization you can generate on the dendrite. And so we look at the blue site, so we're moving away from the chemical synapse a little bit right here, and we see that it does decrease a bit. It's delayed through time, but it's still a pretty big depolarization. And then we look further along in green, and if that is sufficient to drive the voltage-gated sodium channels to threshold, then boom, we get an action potential. All right, so this is known as temporal summation. It's just sending a train or multiple action potentials in order to excite the membrane to a higher level than just a single action potential is capable of on its own. And this is all with the aim of driving the membrane at the axon hillock, the spike initiating zone, to threshold. Now, of course, we also have the possibility that it's not just one chemical synapse that's at work here, but we have multiple chemical synapses. And so that introduces the mechanism of spatial summation. So in addition to this temporal activation, we can have signals from multiple dendrites. So in this case on the left, we see two excitatory synapses simultaneously activating the cell. And let's just say they're both small, and we've moved our microelectrode over, so we're now monitoring this other dendrite here in blue, and we can see that they're both small, but they activate at about the same time in different places, and so that provides two inlets for sodium to move into the cell. And if you look at the green site, you can see that they can sum in a way that creates a larger depolarization than they could generate on their own. And again, in this case, we're above threshold. That creates an action potential. Does that make sense?
Just want to clarify. So with the multiple presynaptic cells, the potential gradually increases based on the chart.
Say that again?
Based on the chart, with the multiple presynaptic cells, would the graded potential increase based on time or distance?
Well, so the depolarizations increase through time, and the two depolarizations sum so that you get a greater depolarization than you would just from one of those chemical synapses being activated. Okay, so in addition to this spatial and temporal summation that's all acting to excite the cell, you can also have presynaptic cells weigh in to try to prevent the generation of an action potential. So we already talked about how you have inhibitory synapses. Now we have the dendrites integrating both excitatory stimuli and inhibitory stimuli. So in our example here, we'll have the left dendrite have ligand-gated chloride channels and therefore creating a hyperpolarization, which we'll see in purple here. Whereas in red, we have a excitatory synapse and a depolarization. Now the chloride channels create a hyperpolarization. Those two kind of sum, or the difference between those two, causes a very mediocre and sub-threshold depolarization, which in this case is below threshold, and thereby prevent the signal from being propagated. So the presynaptic cell shown in red here is trying to drive this cell to threshold to create an action potential, and this one is just blocking it. It's inhibiting that from happening. So, so that's sort of how the decisions are made, through temporal and spatial summation among the dendrites. We have both inhibitory and excitatory signals to that aim. And now you can begin to build networks of neurons that can influence the decision making that is made by a particular cell. So in this example, and as we talk about organ systems, we'll see these kinds of networks as they're connected to organ systems. And we'll see more sort of explicit physiological examples. I just want to lay out some possibilities here just by way of illustration. So here we have one postsynaptic neuron that's receiving signals from multiple, 3 in this case, presynaptic neurons. And we have an excitatory synapse in the case of that cell, and then that one as well. And we might simultaneously have an inhibitory synapse that's going to weigh in on whether or not an action potential is generated or the signal is propagated in the postsynaptic cell. In this particular case, the excitatory cells win, and so we have a decision made that these are more sort of influential neurons than the inhibitory one. So we have the facilitation of the integration of signals from multiple sources. Conversely, you can have a situation where one cell branches out. The axons are capable of branching and activating multiple targets. And so that provides the opportunity for the amplification of a signal. So here we have an excitatory chemical synapse that activates one presynaptic neuron that branches out and activates multiple postsynaptic neurons, which may then innervate effector organs. So think, for example, muscle cells. If you want to activate a muscle, if you just activate a single cell, it doesn't generate much force. But if instead you want to generate the whole muscle, then it's helpful to take your signal and amplify it in a way like this. It also follows that we can, if we inhibit our presynaptic neuron, we can shut down this whole network. And that's what we see here, where we have an inhibitory signal, which then shuts down the network. So all downstream targets are inhibited in this case. This is just 2 examples of how once you start building networks, then you can have much more complex behavior. So on dendrites, we've seen how they receive signals that transduces a chemical signal into an electrical signal, so we get changes in the membrane of the dendrites. They can be both excitatory and inhibitory. At the axon hillock, we have, or the spike initiating zone, we have this decision made about whether an action potential is generated, and that provides the opportunity to integrate signals, both temporally and spatially. So with that, I'll just conclude by reminding you that we have our first midterm on a week from Monday. So you definitely want to review your notes, and we can look forward to that. So I'll see you next time.